Hello friends, welcome to my channel Botanical Studies. In this video, we will discuss about prokaryotic gene expression with the example of LACOPERON and TRYPTOPHAN OPERON. OPERON and prokaryotic gene expression. OPERON is a group of prokaryotic genes with a related function that are often grouped and transcribed together. The OPERON has only one promoter region for the entire OPERON. It is also known as polycistronic. For example, the bacterium Escherichia coli contains a number of genes clustered into operons and regulons. The LACOPERON, which is involved in lactose degradation. The tryptophan operon, which is involved in tryptophan biosynthesis. And the his operon, which is involved in histidine biosynthesis. These operons are turned on when the gene products are needed. Operons were first identified as a mode of gene expression control in 1961 by Francis Jacob and Jacques Monod. Let us study the composition of operon. An operon is composed of structural genes, promoter, operator, and regulator gene. Structural genes. Structural genes that are related used in a biochemical pathway. Promoter. Promoter contains the nucleotide sequences that can bind with the RNA polymerase to start transcription. This sequence also contains the operator gene. Operator. Operator contains the nucleotide sequences that can bind with the repressor protein to inhibit the process of transcription. Regulator gene. This gene produces a protein called as repressor that can inhibit the transcription of an operon by attaching with the operator. The LACOPERON is also known as inducible operon. Let us study in detail. An inducible operon is usually off. In the absence of an inducer, the operator is blocked by a repressor molecule. When the inducer is present, it interacts with the repressor protein, releasing it from the operator site and allowing transcription to proceed. Example LACOPERON. The product of the LACOPERON produces enzyme necessary to break down lactose into glucose and galactose. This allows the prokaryote to use the disaccharide lactose as an energy source when necessary. Structure of LACOPERON. The LACOPERON is made up of three structural genes, LAC-Z, LAC-Y, and LAC-A. LAC-Z makes beta-galactosidase, an enzyme that breaks lactose into glucose and galactose. LAC-Y makes permease, which increases cell permeability for lactose. And LAC-A makes trans-acetylase, which transfer acetyl group. Promoter gene, LAC-P. It is present in the promoter region and act as a binding site for RNA polymerase. Operator gene LAC-O. It is present in the operator region and act as a binding site for the repressor. Regulatory gene LAC-I. Located immediately adjacent to the promoter region, LAC-I encodes for the synthesis of an allosteric repressor protein. Gene expression of LACOPERON in presence of lactose. When lactose and its isomer allolactose are present in the cell, the LACOPERON is to be turned on. An inducer molecule allolactose inactivates the repressor protein. Allolactose will bind to the allosteric site on the repressor protein, changing its configuration and rendering it inactive. Due to this, the repressor protein cannot bind with the operator, so RNA polymerase can able to bind with the promoter and transcription can occur, and the three lactose degrading enzymes can be synthesized. Gene expression of LACOPERON in absence of lactose. The expression of the LACOPERON is controlled by the regulatory gene LAC-I, located immediately adjacent to the promoter region. LAC-I encodes an allosteric repressor protein. In absence of lactose in the bacterial medium, the repressor protein is become active and binding to the operator site. This prohibits the RNA polymerase from transcribing the operon. That keeps the LACOPERON off. Tryptophan operon is the repressible operon. The purpose of tryptophan operon is to synthesize the amino acid tryptophan when needed. The tryptophan operon is an example of a repressible operon. It is always on unless turned off by a repressor protein. The repressor protein is synthesized by TRPR gene, while the repressor protein is always present in the cell, but it is in an inactive form. When a co-repressor tryptophan is present in the bacterial cell, the repressor protein becomes active and the gene expression remain off. But in absence of tryptophan, the repressor protein become inactive and the gene expression will on. Let us study the structure of tryptophan operon. The tryptophan operon is made up of a promoter gene, an operator gene, 5 structural genes and regulatory gene. A promoter gene TRPP, it is present in the promoter region, which is the binding site for RNA polymerase. An operator gene TRPO, it is present in the operator region, which is the binding site for the repressor. 5 structural genes TRPE, D, C, B and A, genes that encodes enzymes for tryptophan synthesis. Regulatory gene TRPR. The tryptophan operon is regulated by the regulatory gene TRPR, which encodes for the synthesis of repressor protein. Let us study gene expression of operon in absence of tryptophan. When the bacterial cell does not have tryptophan, the repressor protein is inactive. So, RNA polymerase can easily attaches to the promoter region and transcribe the gene necessary for the synthesis of tryptophan. That is, the gene expression of the operon is on. Let us discuss the gene expression of tryptophan operon in presence of tryptophan. When tryptophan is present in the bacterial cell, it represses the operon or inhibits the transcription of the operon by activating the repressor protein. Tryptophan is also called as a co-repressor as it attaches to the repressor, which changes the configuration of the protein in such a way that it can bind to the operator and block transcription by preventing the binding of RNA polymerase to the promoter. In this way, the cell saves energy by stopping the production of tryptophan when already present in the cell. So this is all about prokaryotic gene expression with the example of LACOPERON and tryptophan operon.